Hi, everyone. Welcome to Cancer Diagnostics Enabled by Machines. I'm happy that some of you came to the last session today, and I will explain to you a little bit how we try to combat cancer with deep learning. So we were the first to bring actually an AI into pathology routine diagnostics in the United States. And that's quite a big achievement, given that we're a small team of like 25 people. And there are competitors in the US that are like 10 times bigger. So I want to talk to you a bit about how we managed to do so and what were our learnings on the way. But first, a bit background. So I talked to a couple of you on the conference already, and the question I often got, pathology, but why do you diagnose the dead people? But in fact, pathologists, they don't only work on dead people. That's only a small part of their work. They actually do cancer diagnostics, where when someone has the diagnosis, the potential to have cancer, a small piece of tissue is sent into a pathology lab, and there it's cut into thin slices and then stained with chemicals, usually to illustrate specifics about, for example, the cell nucleus or the membrane and things like that. Then a pathologist, a doctor, examines this tissue and makes the diagnosis, which then determines the treatment, so whether the patient will receive specific medication like a chemotherapy, or whether they will undergo a surgical operation, something like that. This would all be great if it weren't for this. So the number of cancer cases is rising more and more. On itself, it's good because that is due to people becoming older and older. And as you get older, you tend to become more often also sick with cancer. However, we also have more complex diagnosis nowadays because more and more individualized treatment exists. But to make the treatment individualized, you also need to have more steps in the diagnosis. You need to be more individual in how you detect what the patient really has, what kind of cancer, and how rapid the growth is and stuff like that. And if your diagnosis is more complex, of course, you need more time. Now we have more cancer cases that need more time, but the number of pathologists is actually declining. And of course, that gives a bad gap, right? Imagine you went to the doctor and they say, maybe you have lung cancer. We'll send some piece of tissue in and let's just wait 3 to 6 months, then we get the result. Doesn't sound so cool, right? So we want to change that, and we want to help close this gap by automating the pathology lab. And when I say automation, it's not necessarily complete automation. It's usually diagnostic support. So we build tools for the pathologists to be faster in their work. And by that, they can increase the throughput and get more stuff done. And here are 2 examples. We have automatic tumor detection, where we look at the large tissue pieces and try to find where are the areas of actual tumor areas. But we also do things as instant cell quantification, where you actually need to find all the individual cells. You need to count them, what kind of type are they, and based on the different counts, you derive diagnostic scores. And I'm really proud that just yesterday, actually, we passed the record of having 20,000 patients diagnosed with our solutions. So we're deployed in 17 labs already in 6 countries. And more people are using this, so that's really cool. But there are also a lot of challenges for AI in digital pathology. And I assume most of you are not very familiar with pathology, or is anyone here pathologist? Okay. So first challenge, it's an extremely difficult vision task. And if you look at this, for example, the upper row, these are what we call mitosis. And if you still remember from your biology school, it's when a cell is dividing, then we call it mitosis. The cell is growing, it's duplicating. And on the bottom row, you see little things that to most of you probably look exactly the same, but in fact, they're not mitosis. So they are kind of artifacts that look similar. And a doctor can, based on certain features and relative position and stuff, determine what is an actual mitosis and what is not. And of course, the AI needs to do the same. There's also a huge variance in clinical routine. So it's not that, you have a picture of a dog and then another picture of a dog and it looks quite similar, but rather each lab does their own work of preparing these images. They do their own chemical staining, and it's a process that's messy sometimes. It depends a bit on the temperatures you're working at. And these are all images made on breast tissue for the staining called Key 67. And as you can see, they look drastically different. For example, lab 3 looks super dark, or lab 4 looks quite light. Some have very dark, almost black colors, some more brownish. And for a human, that's okay-ish because you can just, kind of filter out this difference. But for a machine, it's really difficult because you work on these colors transform to numbers and everything changes. Also, these images, they are humongous. If you print them out, you have a billboard that you can look at. So quite nice advertisements outside. They are sized in gigabytes, and 100,000 times 100,000 pixels are typical dimensions. So also from the computational side, it's a challenge. And just to give you a little example what a pathologist works on. So these big images, they are pyramidal images. You can zoom in and out of them. And they actually need to look for tiny structures, such that I have highlighted here in the red. And they need to detect these enlarged cells there that are actually tumor cells. And now when you consider how small that area is compared to the huge tissue amount, it's kind of like a needle in a haystack problem. So how did we at MindPeak tackle those challenges? The first one, the very difficult vision problem, we solve with different deep learning models. And it's not a single model, actually, because we build a pipeline of models that run one after another. So the first model detects the tissue, that is just background versus foreground segmentation. Then we have the second model that segments the found tissue into what is kind of anomalous tissue, what is probably cancerous, and what's normal healthy tissue. And then the final model, the cell classifier, runs on these detected potential tumor areas and does the single cell classification. And this last model needs the highest magnification, so it's the most computationally expensive to run. And that's why this cascade kind of helps you to make this more efficient, so you can focus the most intensive workloads only on some parts of the image. And we do so also with a lot of different training approaches. So for the large variants, of course, on the one hand, we use augmentations. So we tweak the images we train on a little bit to make them look different according to what I've shown you before in this variation. But we also employed techniques such as unsupervised learning. So typically in deep learning, so who of you is training machine learning models, deep learning, anything like that? Couple, okay. So most often you do supervised learning where you have images and then labels for it. So kind of ground truth measures that doctors put on there. So these are the little colored dots here that I put there. Those would be our annotations. But you can also train on the images only, which enables you to train on much more data. However, the question is then, what can the model learn from, right? If you don't have labels, but you can do things such as you change the image a little bit, and then you expect the model that it predicts similar things for two images that look quite similar. And by that, it can still learn to figure out these variances. And so we combine this and have a hybrid model that uses both of these techniques to, in the end, come up with a very robust system. And since we're in the medical space, it's also very important for patient safety to validate our models. So we do large clinical studies where we have a lot of pathologists that do once without AI and once with AI the diagnosis of patients. And then we compare that and have shown that our model is at least as good as these human experts. And that's where you get these CE labels for. So just to illustrate again, here are a lot of variant images. And when we apply our model, it can detect cells on all of these. Now for the how did we tackle huge images? First of all, we just slice them up into patches. So that enables you to isolate the problem into smaller parts. But also, as I've shown you before, this cascade of models also help to make this manageable because you don't need to train the most intensive model, need to execute the most intense models on the whole image, but you filter that before in your pipeline. And something that's very important to me that I always emphasize is that we need to speed up the deep learning development life cycle. And this is because we want to go from idea to implementation to training and evaluating a model as fast as possible. Why? Well, if you take, let's say, a month for this cycle, then you can try out 12 things in a year. But if you take only a week, then you can already try 50 things. If it takes you 2 days, then you can try more than 100. So the faster you are in this process, the more ideas you can try out. And deep learning is experimental, right? You need to try out a lot of different things. You need to tweak hyperparameters and stuff like that. So you want to be fast to try out as many things to get the best model out. And now I want to give you a little bit ideas what we do there and how we improve that. So first, for the idea stage, you can either, just generate ideas out of your head or read some research papers, of course. But something that is quite cool is, I think, if you look at it from a data-driven perspective. So for that, you could, for example, use your validation set or a subset of it to make decisions of what to work on. And then you can categorize the errors you make on those subset images to focus on the ones that have the highest category, the highest errors. So, for example, let's say someone tells you, oh, we need to improve the stroma to immune misclassification. So stroma and immune are just two kind of classes of cell types. And you say, okay, maybe, but first let's take a look. And then you take 100 images and you look at how often does this problem actually occur in them, and you count that. But you also, at the same time, look at other categories, right? For example, the other direction. What about immune to stroma misclassification? Or maybe you find some scan artifacts. So there are problems on the slide where the scanning didn't work well or so. And after you do this process, you see, okay, yeah, I could try to improve it. There are some errors, but in fact, probably spending my time on scan artifacts is much more impactful because there are a lot more errors. And by that, I can probably improve the model more. So using your own data can really drive you in the right direction of what to focus on. Also, when you start a deep learning project, you need to think about how do I actually annotate? How do I collect my data so that I have the best amount of, so the highest quality amount of data, but also how can I enable my annotators to be fast? For example, when you look at single cells like we do, you could either segment them, so you make let the annotators outline the border of the cell, like I've shown here, and that's nice because you know exactly where does the cell start and end. But it's also a lot of effort to annotate like this, right? Someone needs to manually draw these borders. And the alternative is that you just tell them, okay, just put a dot in the center of the cell with the class of the cell, but we don't care about the outlines. And that depends on what you want to do, right? So if you want to exactly know the borders, then of course you need to go for something like this. But if you're just interested, like in a lot of our applications, counting the number of specific types of cells, then you're also fine with this way of annotation. And you can use that because it's much faster. Then metrics. So in deep learning, you always train the model according to some metrics. So you look at how good is your model on, for example, finding the cells, on classifying them, and on other things. And usually you have several metrics. And I really encourage you to always try to strive towards a single metric, because that way, when you work in a team, you can talk about which model actually was better on your target metric. And sometimes it's not so easy to say what is your target metric. For example, in the cell detection scope, we have, first of all, the cell detection metric, where we measure precision and recall, which can be combined to an F1 score. And in cell classification, the same, but this is already a precision recall F1 score for each cell class. So depending, let's say we have 4 classes, then you have already 5 metrics here. So how do you deal with that? And one possibility is to just combine them together in some linear combination, right? So you need to think about, okay, which one is more important, what do we focus more on. And of course, there's a caveat, right? You always need to also look at all the individual metrics in the end, but having a global combined metric helps you to just go to a colleague and say, okay, I reached this score, or so, and then you have a rough value to quickly gauge the potential of your models. Okay, now we have a nice idea and we want to implement it. And we, first of all, need to focus on code quality, I think, because with high quality code, you are much faster in changing your code. When your code quality is, the code, you read the code much more often than you write it, right? Usually write the code once, and then you read it maybe 10, 15, 20 times over the lifetime of the code. We also automate a lot with CI pipelines and use profilers for CPU versus GPU interplay. So when you load your data, it's often on the CPU, whereas your training is on the GPU, and you need to shift data back and forth. And also other things like augmentations can be done on the CPU or the GPU. And with the Nvidia profiler inside, for example, you can really figure out where during the course of your script are the different systems used, and then you can try to make the GPU as efficient as possible, because you don't want your GPU to idle around and have the GPU process a lot and the GPU is just waiting for it to finish. So some examples for code quality, what do I mean by it? For example, if you have this little function, the loss function, and you want to read it, it's fine. It has a comment, filter out background targets in the top there. So that comment helps you maybe figure out what it does. But I would always encourage you instead to make such an explainable comment as a little function. So that way the function also explains what it does. You can immediately see that. And if you read the bottom code, then you don't need to dive into the details unless you want to. You can jump to that function, of course. But that way the comments don't get stale over time and stick around in your code. And you can change the code and the comment still lives there somehow. Another example is the cool INOPS library. So that's very, very helpful in deep learning, actually, which I will illustrate here. So we have a little piece of code called flatten by channel. And it's a bit messy at first look. It first gets from the shape of the prediction, the batch size, channel's height and width. And then there's some permutation going on. Then there's this contiguous view. And so, you probably take a minute or so to figure out what exactly is going on here. And now compare this with this. You just have, I want to rearrange my prediction from batch channel height width to the combination of batch height width and the channel in the end. So it's much easier to read. It just saves you a minute. And that minute, it saves you every time you read this piece of code or you want to touch it. And I really would recommend using something like this whenever possible. For us, the rule is whenever you touch code, you try to make it a little bit better, since that helps everyone downstream. Okay, most of you probably use CI pipelines for sure. Nowadays, they're very common, but just a little bit, because in the deep learning space, it's not always the same as in normal software development. We still use CI pipelines as much as possible. So for code formatting and stuff like that, of course, for unit tests, even though not necessarily 100% code coverage is there, but for sure the most important things like metrics, losses, transforming data and stuff like that should be covered by tests. Because if you fail there, then it's super messy. You will find it out maybe very late in your project, and that costs you a lot. We also build our Docker images in the CI pipelines and automatically deploy when someone changes code, so you can immediately also inspect what the model does. Because the earlier you find bugs, the better. And of course, all of us make a lot of mistakes. So it's great to have some trap guards that help you there. Then for reproducibility. So if you consider this cycle, you also want to be sure that you know exactly what was going on. And nowadays, everyone uses Git for tracking your code, right? And DVC is called data version control, is a tool that you can use to kind of couple your code with your data. So you check in DVC files, they're like little metadata files that you check into Git that determine exactly what kind of data was used when you had that code state. So later, if you want to, compare some new model with some old model, you can still check out your Git state that the old model was trained on. You know exactly what data that was. And then you can also train the new model on exactly the same data, because otherwise you don't know, is my new model better because I trained it on more or new data, or is it better because I implemented some new ideas? And to kind of differentiate that, you definitely need this. And it also brings you cool other capabilities like this pipeline. So you can make pipeline steps that depend on each other. Like here we have the raw data. Then we generate our training data from it, compute some statistics, then run our training, and finally the test set evaluation. And the cool thing about that is, let's say you change something about your training pipeline, so in the run training step, and you rerun, then DVC will figure out, okay, there was only a change in that training box, and before nothing changed. So I don't need to regenerate the data, I don't need to recompute statistics, I will just rerun the training and the evaluation. And that way, again, you save a lot of time and can be much faster, because generating the data can take hours or days. Okay, we had our idea, we implemented it. Now let's train a model. And this is typically what happens. It takes a lot of time to train a model because it runs on the GPU. You have a lot of data, and you need to train on a lot of data to get it good. So what can you do? You can speed it up by training it on multiple GPUs. And you've heard everything about ChatGPT and Co. They train on super many GPUs. And I will talk a bit more about that later. You also can reduce your data set. So you can say, okay, I make a toy data set that is representative for my problem as far as possible, but I can use it for fast experiments to then only run my most promising ideas on the large data set. So let's take a look at multi-GPU training. There is in PyTorch, so we use PyTorch for most of our deep learning code, and there's something called data parallelization, where you can split up your data onto multiple GPUs. So instead of having the single GPU on the top that trains one batch at a time, you can say, okay, I'll train batch 0, 1, and 2 in parallel on 3 GPUs, and then I do that step by step. So theoretically, I would get like 3 times faster training, right? Not quite, because of communication overhead. There's a lot of overhead in this process, because first of all, the master GPU prepares the data and model weights and sends that over to the other 2 GPUs. So that's the first communication. Then each GPU processes a batch, does the normal calculation. Then they need to gather the results back at the master GPU. And finally, the loss is scattered back to all the GPUs, and then the gradients are computed on each GPU. And as you can see, almost every step involves some communication. Even in the end, we still need to update, of course, the model again. So another step of communication. So that is not so good, but instead there's something better. It's called PyTorch distributed data parallel. And I really would encourage everyone to use that. I think nowadays also the instructions say so that you should prefer that one. So how does this one work? First of all, on each GPU, a separate batch of data is loaded. And I'll get to how that works, because it might be a bit magical that it just works. And I'll get to that. Then the loss and the gradients are separately computed on each GPU. Then just the gradients are communicated. So this is the only communication step here. And finally, the same model update happens on each GPU. And this is because they all started with the same model weights. They only communicated all the gradients. So in the end, they have all the same information. They started from the same point. They have the same information. So when they update, they all are at the same point in the end. There are a little bit of things to mention. So first of all, how do they load the data? You need to have a special sampler for that. So your sampler takes care of making sure that not the same data is repeated, because you don't want to end up with having the same batch on 2 GPUs. There are risks for overfitting and stuff like that. And you can use either the distributed sampler that PyTorch brings per default, or you can extend it and make it to your custom solution. And I also want to mention the cool paper from Goyal et al. It's from 2018. So if you do multi-GPU training, you should consider this, especially learning rate warm-up and scaling, because otherwise your performance is not as good as training on a single GPU. Okay, now let's talk a little bit about disappointment, because often you end up in a state where you think you have a cool idea, you read a cool research paper, you implement it, but then it doesn't work, right? It doesn't help. Your baseline still is better, and you're kind of frustrated. But I think that's normal in machine learning, so you need to iterate a lot. You often fail, that's normal. Ideas don't matter so much. It's more about measuring and improving over your metrics and seeing where can I actually improve. And even when you fail, there's always an opportunity to learn from it, to ask yourself, why did it work in this paper but not in my own implementation now? What can I learn about my data, about my problem? Can I generate new ideas out of this failure? And that way you learn and improve even from your failures. We also always write up diaries or little blog entries. We use Confluence for that. You can, of course, use whatever you like. And that way is also cool, because when you later have similar ideas or problems, you can look up what happened before or what your colleagues did. And when you have high speed in your cycle, then you can always generate just the next idea, because it just takes you a few days or so. So to sum up, I think when you can, you should always use your data to drive your idea generation, to kind of let your idea guide you in the direction where you can improve your models the most. It helps to have a single metric to do this kind of guidance, but it's also useful to look at detailed metrics for sure. Automate as much as you can, because the more you automate, the less time you spend on those things for sure. And don't only track your code, also track your data. DVC, we have very good experience with that. There are a couple other tools out there as well. But yeah, we're really happy with it. Make your code quality as good as possible, because that way you help your team and you have the virtuous cycle of being faster and faster. And when using multi-GPU training, always use the distributed data parallel option rather than the normal one. So, and last but not least, as I said, when you fail, when there's something's not working out, use it as an opportunity to learn from them and to make your models better in the future. So overall, just iterate quickly, have fun doing it, and train a cool model. That's it from my side, and I'm looking forward to your questions. Thanks.